with ERPs or functional MRI or so forth. Is that clear? Okay. On the other hand, if we want to know this kind of causality from some neural activity we measure in the brain to either a behavioral response or a subjective feeling reported by a behavioral response or something like that, that's the challenging part. That's the kind of causality. Okay? So last week we talked about a bunch of them, and today we're going to wrap this up talking about TMS and animal studies. But first I just want to remind you very briefly, I won't go through in excruciating detail, we talked about behavioral methods, which are great for characterizing internal representations, as you saw with face inversion effects, and some of the other behavioral data. Its ability to tell you whether the neural activity you're looking at is causally involved in behavior is like nil. Okay? Now, at least one of you, I only read a few of the assignments, but at least one of you was a whole causal chain there. Now let's consider what kind of causality we're talking about. There's one kind, which is that the stimulus causes neural activity in the brain. That's a kind of causality that we can absolutely test, even if we're measuring that with functional MRI, because we can mess with the stimulus. A little bit more about that temporal resolution. I know I kept saying the temporal resolution of functional MRI is lousy, but I had run out of time and skipped through the key slide, so let me back up and do that here. This is the bold or MRI response as a function of time. This is an idealized version of it, but it looks kind of like that. Suppose that we stuck an electrode in my primary visual cortex, and we flashed up a very brief visual display. Okay, here's a visual stimulus on for maybe 1/10 of a second, bright flashing thing V1 loves. V1 also primary visual cortex, right? I know that from work in animals and even some work in humans. Okay, so super fast after the stimulus. Okay, it just goes straight up from the retina to the LGN V1, boom, there it is. So all the neural activity happens right there and ends right there. But the MRI response. So what's relevant here is not just that it's delayed, but it's big and sloppy, right? And so both of those things are the reasons why functional MRI responses aren't good for distinguishing what happens on a fine temporal scale of, say, events less than a second. I showed you with that amazing movie of the guy getting stimulated in his fusiform face area and seeing percepts of faces on top of whatever he looked at. That's a kind of quintessential, beautiful example of the causal role of neural activity there. We're basically directly manipulating neural activity. We're injecting neural activity there electrically and looking at the trouble. But mostly people don't stick it back there, and actually the subjects won't let you anyway because there's a lot of neck muscles, and it really hurts when you do TMS over muscles. And so if anybody had such a stupid idea as to try to zap the brainstem, the subject would probably like object immediately before they got very far with it because it would hurt. And you guys are all probably wondering, how safe is this? It's not totally clear. There have been lots of studies in animals where they, zap a rabbit, 100,000 times or something like that in a second. Okay? So here's what David Pitcher found in that study. This is accuracy at this same different matching task. When you stimulate the right occipital face area versus vertex, that means you stick the coil up here, which is pretty far away from face regions. It's kind of a control condition, not a perfect one, but better than nothing. By the way, TMS usually doesn't hurt unless you stick it over muscles. You stick it over the frontal lobes, and I don't know, every time I try to disrupt my language abilities, it hurts too much because there are muscles up there. But most places, like the top of the head, there aren't muscles, and it doesn't really hurt. But it still makes a loud cracking noise, and it's kind of like somebody went like that. And so you'll get electric fields perpendicular to the coil, sticking right into the brain like that. And what do you think happens if you stick a big, huge, transient electric field, boom, into your head like that?
Is not a magnetic field.
Yeah, you're right. Right-hand rules magnetic field. I was thinking I was misremembering, right? Electric current makes magnetic field, right? Anyway, for current purposes, it doesn't matter. Either would do it. Actually, there's a variant of this where it's an electric field, but it's debated how well that works. Okay. Anyway, what happens is you affect neural activity and tissue right underneath the skull, right? Okay. So all right. So then I talked about studying patients with focal brain damage. And here you really can make a strong causal link between a bit of brain and a behavioral ability. You lose that bit of brain, you can no longer do that task. That's a patient who can do B but not A, say recognize faces but not objects. And when you have in the literature 2 cases like that, now you're in a really strong position to infer that there's something fundamentally different about face recognition and object recognition in the brain. Okay, so that's really important. It's a very interesting, complicated one. I think it will ultimately have to do with how those batches of neurons not only respond to faces or light or whatever, but how they code for that information, such that when you put a big kind of artifactual, non-biological signal in there, will it have any meaning that the subject can interpret? They popped a monkey in a scanner and did the very same experiment that we do on humans. Okay, so here's a monkey brain. Again, the cortex is unfolded so you can see the whole surface. The dark bits are the bits that used to be inside a fold. The little yellow patches are the patches that respond more to faces than objects. Just analogous to the FFA in humans, but there are 6 little patches in monkeys. Okay, so that's kind of, so far that's like, okay, fine, monkeys have them too, that's cool. But the thing is, because it's a monkey, you can then stick electrodes straight into that region right there. And you can record from hundreds of neurons in that region. And you can record the response of each of those hundreds of neurons to hundreds or thousands of stimuli. You can characterize the neural code. I'm going to take a moment to talk about ethical issues in animal research, just to note that I think there is an issue. And I'll say that it's not unreasonable if you have qualms. I noticed in an earlier lecture I started talking about recording from animal brains, and I didn't have time at that moment to mark this, but I do think it's important. If it makes you uneasy, and I think there's a very difficult trade-off between avoiding suffering in animals and research that has saved countless lives. People can legitimately come down on different sides of this, but many lives have been saved, including mine, based on animal research that enabled treatments that were life-saving. Code seems to be saying here, and then here, and then here, and you can watch that those codes change over time in each of those regions, and you can see different representations in each of those regions. It's quite breathtaking. You can answer this question of what are the anatomical connections between these regions with a whole bunch of different methods that I won't go through here, but you can actually answer what, what's connected to what, and what these guys have got this. But let me just say, prediction is supposed to be extremely precise. It's the exact statement of what you will see when you measure your dependent variable if the hypothesis is true. What is the crucial thing you have to look for in the data you measure that tells you if the hypothesis is true or not? And the prediction is what you the problem. If I told you, okay, look at my face and don't process, low-level visual information and don't think about what I'm saying, just see my face. It's like, what? You can't do that, right? All right. So what you aspire toward in the contrast that you choose is something called a minimal pair, right? So the idea is we're going to have these 2 tasks that are identical in every respect, except for that one thing we care about. Okay, so here's a task. That's good. That deals with the grass confound problem, right? Absolutely. Very good. What else? David, you had interesting ideas.
Well, we were talked a lot about animate versus inanimate things, so like comparing it to a garden hose.
Yes, garden hose. Love it. A few examples. What were some of the what were some good kinds of control conditions, that is non-snake stimuli that are good to compare to snakes, that maybe aren't perfect minimal pairs, but that get partway there? I saw a few just in the few papers that I looked at. Yeah, I'm sorry, I've asked your name like 6 times, but I'm going to, on my trusty sheet, tell me again how you say it.
Eke.
Eke. that snakes have, and it could be that that's the cue. So then the trick is you have slithery motion versus what? I don't know. That's hard. What other kinds of motions could you have?
Right, so you could have just different rigid motions where it's not slithering, or it's not like changing shape.
So one idea was to have them, like essentially indicate if they saw a snake or didn't see a snake, but that brought up problems like, well, we don't want them thinking about snakes the entire experiment. So
Also, if they're going to tell you all the other stuff, like, how you arrange your stimuli over time and all that kind of stuff, okay, it matters a little bit, but this is the crux of the matter. What are those conditions, and are they the right kind of minimal pair? Okay, and you guys got the gist, but I felt like most of you didn't really engage. Okay, what exactly were those non-snake conditions, right? So that's really the crux we're going to go. Are you going to assign different conditions to different subjects? So these 5 people will see all the snake images, and these 5 people will see all the non-snake images? Or are you going to have snakes and non-snakes within each subject? And what are the trade-offs there? And what is the order of conditions? Okay, within a run. And we won't get to number 10 for the moment. Okay, so we're going to break you guys into 4 groups, and you're going to talk amongst yourselves and try to come up with your best answers to these. About that. What I meant by a baseline is different from a control condition. A control condition would be like the non-snakes contrasted with the snakes, right? Baseline tends to be like a minimalist condition that's supposed to, turn the brain off. Okay, can we turn the brain off? No, of course not, right? But we can aspire toward it. We can go far. Okay. So you could just have a blank screen. That feels like a pretty minimal thing. You could have people close their eyes. The reason that in vision experiments people tend to have fixation, where there's a tiny dot and subjects are supposed to hold their eyes on it, is that in natural, left to their own devices, people move their eyes a lot, several times a second, and moving your eyes produces all kinds of activity and lots of neurons. And so it's a very kind of active visual thing, even if there's nothing on the screen. And so staring at a